he was trained as a biochemist at the University of Copenhagen and at the Carlsberg Laboratory, and then completed his PhD at the University of Cambridge in 2004 under the supervision of Professor Christopher Dobson. He then moved on to be an assistant professor in Copenhagen before joining the D.A. Shore Research Group in New York in 2007. In case you don't know, that's the Anton computer, among other things. He returned to Copenhagen in 2011, where he now serves as a professor for computational protein biophysics at the Lindstrøm Lang Center for Protein Science. He received the Danish Independent Research Council's Young Researchers Award in 2006 and was a co-recipient of the 2009 Gordon Bell Prize and has received several prestigious grants, including the Hallas Möller stipend in 2011 and another starting grant in 2012, which I cannot pronounce. And most recently, Novo Nordisk Foundation Challenge Program grant in 2019. He's the director of the Lundbeck Foundation Brainstroke Initiative in Structural Biology and the Novo Nordisk Foundation PRISM, Protein Interaction and Stability in Medicine and Genomics Center. His current research interests include developing and applying computational methods for integrative structural biology and the integration of biophysics and genomics research. As an addendum, I encourage you to follow him on Twitter because he always has something. He always has something interesting to share, from specific simulation stuff to very nice divulgation. I don't know how to say it in English. Anyway, very nice books about biochemistry written, for example, by Nick Lane. So, without further ado, thank you very much for being with us, Kristen. The floor is yours.
Thank you very much for that introduction, and thanks for having me here, even if it's only online. It's a pleasure to be here and to tell you about some of our work. And I'll just get the laser. No, actually, I'll just minimize the window here. Sorry, and then I will get a laser pointer. Now I can only see myself, so I'm sorry about that. Yeah. I've been going to real proper physical meetings, so now I can't remember how to move things around here. Height floating meeting controls. Things are slowly getting there.
If anything, we can see you. There you are. Good.
Yes. All right. So yes. So I'll talk about the work that we've been doing using simulations to guide and interpret experimental measurements. And as Nina mentioned, a lot of our work is supported by various Danish foundations. In particular, a lot of the work that I'll talk about today is supported by one of the private foundations in Denmark called the Lundbeck Foundation that supports research broadly, but in particular in the area of natural sciences and neurosciences, but also biophysics broadly and structural biology. So I'll talk about work done in this Brainstroke initiative that was just mentioned. And sort of, I tried to be a little bit ambitious in the terms of the areas that I wanted to cover, which means that I won't cover all of these in any substantial detail, but to give you some ideas of the different kinds of things one can do when using molecular simulations in the context of experimental measurements. So the first, probably third of the talk, will deal with how we can use molecular simulations to study protein dynamics. Mostly in the absence of experiments, but in ways that we can then benchmark or compare with experimental measurements. This is often useful if you don't have detailed experimental data, or if you are trying to study things that are very difficult to study experimentally. And then in the second part, I will talk about how we can more directly incorporate experimental measurements of various structural and biophysical types into simulations. So I'll talk about the work that we've been doing by integrating it very directly into sort of system-specific ways, talk about this Bayesian maximum entropy method that we are using and give some specific example on a nanodisk. And then I will talk about how once you have enough examples of things that don't work, and we have many examples of things that don't work, how you can use that information to modify or refine or even fully parameterize various types of molecular force fields, in particular in the context of coarse-grained simulations of flexible or disordered proteins. Before I do that, I'll just give a reminder to myself and to you on why we do simulations and not just experiments. And I'm illustrating it here with a sort of simple picture that's not about molecules, but I hope will illustrate the point that I will make in a few slides. And this is that we care about the structure of objects. This is a structure of a horse, but we also care about the structures of biomolecules. But we also are interested in understanding how these things move and change their shape. So if we want to understand biological processes, we want to understand how do things look and how do things move. And if you are a very skilled experimentalist and have systems that are well behaving, you can sometimes resolve experimentally many different states along some specific pathway. In this particular case, we can sort of see the horse sort of both in its ground state here, but we can also sort of watch the actual process, in this case using the technique that you're all familiar with, photography, but coupled in this case to sort of a high-speed camera and also sort of stroboscopic light that enables you to sort of really resolve the system both at high spatial resolution, so the pictures are sort of very detailed, but also high temporal resolution, meaning we can sort of see snapshots along the way. So this is the ideal case often in the experimental world, but unfortunately reality is often very different. And this is illustrated in this picture that I fell in love with the first time I saw it. It's the same technique as before, it's photography, but it's different in several different ways. First of all, that it's no longer a horse, but a swimming pool. Which is just the object of the picture. But there's also other differences. So now you can see you're not resolving all these different states. Instead, there are other sort of strange things about this picture. And without sort of spending too much time on it, one thing you might notice, if you stare at this picture long enough, is that there's something odd about the lighting. And what you will sort of notice is that there's sunlight coming in sort of apparently both from this side and from that side. And the reason for that is that this is a picture that's been taken with what's called a pinhole camera. So with a very small hole, which lets in just a few photons at the time. And the way to compensate for that is that you have a very long exposure time. So this picture was taken sort of with an exposure time of about half a day or so. So this is very different from the previous example with the jumping horse. Right here, we're averaging over a very long period of time. And when you do that, you start to get artifacts in your experimental observation. In this case, obviously, there are no two suns shining down here. There's a single sun that moves relative to the swimming pool so that it appears as if there's sunlight coming in from both sides. What's also sort of surprising is that the water appears completely flat and still. And anyone who's been to the swimming pool in particular, when it's full of kids like it has been here, all these details average out. So the details of the system are not really represented in the experimental data, or at least a lot of the details are averaged out. So obviously, we'll talk later about biomolecules and different types of experiments, but there are a lot of analogies. So when you're doing an experiment and you're recording information about a dynamical system, but your averaging time is much longer than the timescales of the dynamics of the system, there's a lot of details that you won't see. You won't see all the details of things that are moving in the system. What you'll see is a nice average picture of what the swimming pool looks like on average over long periods of time, but not resolving all the details. So the reason why we do simulations and not just experiments is then in principle, by simulations, you can see all this dynamics, right? You can see the swimmers moving back and forth. You can see the sun and the earth moving relative to each other. And so you can, if you have a good model of, rotation of earth and the sun and swimming pools, you can simulate this. And then you can say, I'll do a calculation on the average view of a swimming pool. And then I can get an image that might look like this, and I can compare that to an experimental average, or in the case of biomolecules, I can compare to average data, for example, from an NMR experiment or small molecular X-ray scattering experiment. And then I can see, do they match each other or not? So when I put down the title of this talk as being whether simulations and experiments is a perfect match, ? There's sort of two aspects of this. One of them is, are they as technologies a perfect match? And secondly, when we do a simulation, does it match perfectly experiment? Often they don't. And what I'll talk about, for a lot of this talk today will be about what you can then do when they don't really match. But the basic outset is that most experiments give you a lot of details about the systems that you're looking at, but there's also a lot of the details in particular about the dynamics that you really don't have access to. So when we sort of think about the interface between experiments and simulations, there are many ways of thinking about this, but sort of conceptually at least, we've sort of divided up the interface in sort of two different ways. You can either do a set of simulations in your computer, and then you can do calculation of observables, this we would call then forward modeling, and then you can calculate the observables. This could be, NMR data, small intellectual scattering data, and then you could do a comparison, and then you can say, well, is this simulation reasonable or not? And then you can make, inferences based on this, and you can say something about the system. Often what we do, and this is what I'll talk about in the second part, is that we start with some set of experimental data, often approached by people abroad or fantastic local colleagues here at the University of Copenhagen. And then we say, how can we use simulation to help interpret this data? And this is then what we would call inverse modeling. But of course, they're tightly related, as I hope I'll talk about later. So in forward modeling, really one of the things that you can do is that you can start really to run simulations of different systems, and by comparing them with experiments, you can get information, for example, about how good the physical models that we use. This is an old slide, and it eventually should be updated. But what I want to convey to you is that the protein force fields that we as a field are using are pretty good. These are examples of proteins that we were able to fold, 10 years ago for most of them. And that revealed that the force fields that we had available at the time were pretty good, at least in some aspects. But then also many other comparisons, and I'll give you some examples in a few slides, show that they're clearly not perfect yet. But the protein forces, at least in the context of studying, for example, protein folding, and can we sort of predict the structure of small, very fast folding stable protein, turns out to suggest that, in many cases you can do that, but a lot of the details and the thermodynamics and the kinetics is, not always perfectly matched. We were sort of then more interested in sort of the more biologically relevant dynamics, and so almost 10 years ago we started sort of on a set of projects where we sort of wanted to ask the question, well, what about more subtle dynamics in the context of native proteins? How well are we doing? And I won't really give you any details, and most of the systems, I'll go through one of them in a little bit more detail, but we sort of found a set of different systems that had nice experimental data, mostly from NMR spectroscopy, but also some crystallography and fluorescence data that we thought would enable us sort of to benchmark how well the simulations were doing. So the first system we picked was a protein called cyclophilin A, where there's really nice NMR data and X-ray crystallography data. So we knew the structure of two different states of this protein. We knew roughly the kinetics and the populations of interconversion between these two states. And then Elena Papaleo, when she was a postdoc in the group, essentially showed how you, by simulation, can sample this conformational change. And actually, we do pretty well in matching this. So at least this was from one example of a quantitative benchmarking of molecular simulations in the context of native state dynamic. We then moved on to a slightly more complicated system. This was work done by Yong Wang. I'll show you his picture in a few slides. He then moved on to a system that had also had nice information. Again, mostly two states populated in this sort of model system. We knew the structure of both states. We then only used one of the states as starting point for the simulations, the sort of the crystal structure, and showed that we can sample the equilibrium between the two different states, calculate actually the equilibrium populations of both states, even estimate the rates of kinetics. And also in this case, the simulations actually did a relatively good job of matching these things. So both of these were relatively successful, but also small conformational changes. And in both cases, we knew roughly what the structures of both states looked like, even if we sort of didn't really put in that information very directly, although it was to some extent. This is described in these papers. So we then moved on to some more complicated systems, and this is one of them, and this is the other one that I'll talk about in the second example. And these things have been published in the last couple of years, and these were much more difficult to study. And as I said, I'll talk about this cyclin CDK2 in a little bit more detail. But again, this is just to illustrate to you that we can actually then run simulations of conformational dynamics of these systems and compare to observables, in this case, populations from NMR spectroscopy. And what the results show that we can, with quite a bit of effort, sample the conformational dynamics, in this case, switching between quite different structural states, calculate the populations. And in this case, actually, the data wasn't as quantitatively good. So we could see the different states, but the populations were actually not super accurate. And so this was already starting to show that there are clear deficiencies in the force field. And, in this process, we changed force fields to the, most current updated ones. So I'll talk about the cyclin case in an example, but in both cases, you have sort of a, this helix shown here in the middle can sort of transiently unbind and come back to do auto-inhibition of this DH domain in VAV1. And as I'll talk about in this ternary system over here, again, you have sort of a similar example where something can transiently unbind and come back, and this is important in the regulation, and we can start to address these things using molecular simulations. So in particular, the question that we were interested in both of these last examples was something that when I found out that this existed was very puzzling to me, but also very intriguing. And the work that I'll talk about is work done by João Enriquez when he was in the group. So over here, sort of the traditional way of looking at this ternary system with cyclin A, CDK2, and P27. And what I'll sort of highlight to you are these two tyrosines here in P27 that are key in this system. So this is a traditional ribbon view. If I now show instead sort of a more space-filling view, you can see sort of what the volume of the proteins look like, and you again see these kind of tyrosines and this sort of nice interface between these two proteins. And if I then sort of don't make the surface transparent, but just show you sort of a normal space-filling surface, right, you can see that you almost can't see P27, and you certainly can't see these two tyrosines. Now, this is interesting because these two tyrosines need to be phosphorylated to activate CDK2. So phosphorylation of these tyrosines enable then, dissociation of this complex, eventually an activation of CDK2. So the question that we are interested in and have studied in a couple of systems is, how do kinases phosphorylate tyrosines when these tyrosines are completely buried, and when this burial is important for the function, in this case, the inhibition of the complex? And many people have looked at this in different ways, and we wanted to see, can we study this by simulations? And I, won't spoil too much by simply saying that obviously protein dynamics is important. So you have sort of an equilibrium between, the buried state of these tyrosines and the transiently exposed state of these tyrosines that the kinase can then access. But of course, if they're exposed all the time, they don't really do the process of auto-inhibition. So this is a very important detailed balance. And what we were trying to do was to ask the question, can molecular simulation do this? And this, I thought, was going to be relatively straightforward. It turned out to be incredibly difficult, both in this case and in the VAV1 case. And I'm just here sort of very briefly summarizing essentially Joao's hard work for a couple of years with the simulations over here on the left. And on the right, I'm showing you the data from an experimental study from Richard Krivakis' lab and with Klaus Seidel also, that did NMR and fluorescence measurement to study the same process in more detail than we did, but with some very comparable results. So the basic thing that we could do is that we could sort of look at these tyrosines, and we can then with various enhanced sampling method look at the transient exposure and the free energy of exposing these tyrosines. So if we look at this system when there's no phosphorylation, both of these tyrosines are deeply buried, but you can then sample the equilibrium. In particular, you can sample the equilibrium where one of the tyrosines, particularly the one that's called tyrosine 88, can get transiently exposed. And this is, you can see down here how it's buried. And eventually this tyrosine might get exposed in this video, and over here it's sort of buried. And, so you can see them going sort of in and out of this system. Now you can see down here how it's getting sort of transiently exposed. Now the kinase can access it and phosphorylate it, whereas this other tyrosine that's sitting up here is just completely buried. Then what we did is that we said, okay, now this tyrosine can be phosphorylated. Let's put a phosphate group on it and then do a simulation comparable for the other tyrosine over here. So now we have phosphorylated this first tyrosine. This is matching this sort of PY88 system. And now we can sort of see transient exposure of this other tyrosine over here. Now you can see that it's out. The kinase can come in, phosphorylate it, and then of course it can't rebind. Or if the kinase doesn't come in, it will rebind. So again, we can watch this equilibrium of transient exposure and burial of something that is most of the time buried, and with a process that is known experimentally to take many milliseconds. So something that would be difficult to study with normal simulations, but with the details that I will talk about with enhanced sampling, we were able to sample these states. And then finally, we can put a phosphate group on both tyrosines and study the double phosphorylated group. We can see that there's this sort of hydrogen bond between this beta sheet between P27 and the remainder of the complex that sort of keeps it together, but eventually this will dissociate, and then other downstream processes will then sort of lead to activation of the system that, I'm not, that we didn't simulate, and I'm not going to talk about. So what we got out is we got some interesting insights about how can these tyrosines be transiently, exposed, what are the interactions that are important, and we could also calculate the populations and compare them to the data from Krivakis and Seidel's labs, and show that there was some agreement, but again, there was not quantitative agreement on the equilibrium constant. So again, highlighting some of the advantages, but also some of the problems of even, I would say, state-of-the-art molecular simulations with enhanced sampling methods and the best force fields that are available. So clearly things are good, and we're getting to the stage where we can start to actually compare to experiment. And sometimes we find that things agree well, like in the cyclophilin A and the T4 lysozyme system before. And in other cases, we find sort of somewhat good but imperfect match between experiment and simulation. Another system that we've worked on, and again, this is work by Young Wen that I mentioned before, is a membrane protein, a system that is difficult to study experimentally. We're starting here, and this is a collaboration with Paul Nissen's group in Aarhus. So they determined the first structure of a P-type ATPase of this particular family that are called lipid flipases, and found the structure of that's shown over here, that we then used as starting point for addressing a couple of different questions that were not so easy to address from in this case the cryo-EM data, which is how do lipids engage with this thing and how are they transported across the bilayer. So the function of this protein is that it sort of will bind phospholipids and then transport them across the membrane bilayer. We're not studying the full cycle. I'll show you, some of the data next time. We'll sort of studying the initial binding and half transport of a phospholipid. And again, to show you the kinds of things that we can do with simulation and some of the things that are difficult to do. So if we just take this protein, we put it in a bilayer, we run standard sort of semi-long time scale all-atom molecular dynamics simulation, we will see quite rapidly binding of a phospholipid to a specific group that was already sort of seen in the structure. Now we can sort of see how the phospholipids can sort of engage in this. So we find a nice binding site for the head group, and you can see sort of the lipid tails over here. And then what I won't talk that much about P-type ATPases, but you might be familiar, for example, with other ATPases of the same family, like Circa, a calcium transporter, or the sodium potassium pump. So these are all ion transporters, and this is the same family. So this is a protein that evolved originally probably to be an ion transporter and sort of has evolved into being a lipid flipase. And the mechanism of this is that there's this sort of charged head group that will then be transported. And the question that we're sort of trying to address is, what do these lipid tails do? They're sort of not part, they don't look like, a normal ion, right? They're just these long floppy things. How are they transported? So we can see initial binding, but the timescales of even moving this thing sort of up to this group are extremely long. So we switch to sort of more coarse-grained modeling. We use a particular model called the Martini model. I'll get back to that later. And one of the questions we're trying to address is, how does this protein achieve selectivity so that it flips some lipids better than others? So when you simulate it in a lipid bilayer that has PS lipids, of course, it binds PS, the same for PE and PC. If you simulate it with a mixture of PS and PC lipids, you can see that it selectively binds PS lipids more than PC. And if you, for example, simulate it with PSP and PC, you can see that there's sort of an order of event of binding preferences to this groove down here that tracks what was then known experimentally about the substrate preferences for this molecule. So again, the molecular simulations can capture some of the things we know sort of from macroscopic experiments, that there is a selectivity towards some types of lipids relative to others. So there's some kind of selectivity, for example, in the head group of the lipids. And we can also see that there are densities, in this case for the head groups, that we can see in the simulations. So we can see that the head groups can sort of occupy different states along this kind of groove going up. And the question we then wanted to address, where does this selectivity come from? Can we see the kinds of interactions that achieve selectivity? So it turns out that even in this kind of coarse-grained simulations, it's actually difficult just by running simulations to see the lipids coming in and out. Again, it's a very slow process. So what Young then did is to turn to free energy calculations in collaborations with others to calculate free energies of binding or relative free energies of binding of different types of lipids. So again, for example, he could calculate the free energy of binding to one of the sites, the deep site we call it, or to the entry site of PS, PE, and PC. And again, not giving you all the details, again, we can find this kind of order of event that the PS binds more strongly than the PE, and the PE binds more strongly than the PC. And there's selectivity both at this entry site over here, but also at this deep site over here. So this whole channel or groove of the protein appears to have some kind of selectivity for certain head groups relative to other head groups. And of course, this is in part due to the charge of that group, but other factors also play a role. So now we know a little bit more about what's recognized. Let's see if we can understand also how this molecule is flipped. So we studied only sort of what I would call sort of a half flip move. This is illustrated with this work here. This is again now back to all atom simulation. This is work by Felix Kummerer. And what he's doing is essentially he's pulling the head group from below the membrane. So in the normal phosphate bilayer, he's pulling it up towards the middle. So that's this purple line that's tracking the Z component. So orthogonal to the membrane plane. He's pulling that up, and then he's asking what's happening with the lipid tails, and this is then the blue and the green line here. And what you can see is that even as he's pulling up the head group sort of from the outside to the middle of the bilayer, the tails sort of stay more or less in the middle. So you start with sort of a standard lipid orientation where you have the head group at the bottom and the two tails in the middle of the bilayer, sort of flipping sort of 90 degrees. So you end up with sort of a more horizontal organization where you have the head group in the middle and you also have the two tails in the middle. So the mechanism of flipping is that you have the sort of the head group being transported up as the ion, and then the tails are just sticking out in the membrane and are sort of effectively, in this case, sort of half flipped. And then something happens that I won't talk much about, and then it's sort of flipped all the way. And what we can also see is that we can see that this head group stays solvated throughout the entire process. This is illustrated over here, but in the interest of time, I won't talk about that. So again, you can see here that this cavity is sort of filled with water that enables the head group to move on. And then eventually it will go to this other side that's sitting on the outside, but you can see that there is a very tight seal between these two different water compartments. So the head group will sort of first move up to this water compartment, and then other chemistry will happen in a completely different domain that will enable a conformational change that would effectively seal off this water down here. And then there will be sort of breakage of this seal, and then the phosphate head group or the lipid head group can sort of move over here and come out over here. And sort of we can sort of, by combining the simulations with different locations and different EM structures and crystal structures, we can sort of see more or less sort of the pathway of how this head group is sort of moving up this flippase. So again, an example of how you can combine simulations and experiments, but again, in ways where we're sort of doing more or less standard molecular simulations, all atom or coarse grain, using experimental structures as input, but trying to sort of come up with interpretations of explaining mechanisms, but without using the experimental data very directly to model the process. So these are, we and many other people do this kind of work, and this can often be extremely informative for studying mechanism. But of course, as I've showed you a couple of examples of, these things are not always perfect. Which then leads me into the second part, where I'll talk about how we can use experimental data more directly to study molecular systems. And in particular, what I'll try to sort of to explain to you is that while there is, in some cases, you can have very large amounts of data and you really need a very simple computational model, you can determine nice structures, or you can have a really nice computational model, and in the absence of experimental data, you can get a lot of insight. But for many dynamic and complicated systems, you can't really do everything by experiment, and you can't really do everything by computation. So there's this whole range of things in the middle where you, with some amount of information and a reasonable but imperfect computational model, can say a lot about the structure and the dynamics of biomolecular systems. And now I'm coming back to this early point about averaging, that I just want you to remind you that the data that we are dealing with in general is averaged over long periods of time and many molecules. We have micromolar, millimolar, nanomolar, depending on the experiment, concentration. So we are averaging over 10 to some large number of molecules. And so if someone measures the average spin of a coin and they repeat the experiment a gazillion times and calculate the average Z vector from this thing, right? It would be 50% spin up and 50% spin down. That would average out, and you would measure an average, a zero component along Z of this vector attached to the coin. So if someone measures, I measured the average Z vector of a coin. Can you tell me the structure of a coin? If I were sort of very naive, what I would do is I'd be rotating my coin, and then I would look for conformations that fit the data, and I would end up with this solution down here, right? This fits the data perfectly. It has a zero Z component, and so it matches the data. But of course, you all, if you've done this experiment, this is an extremely unlikely scenario. And the error that I made was pretty obvious, right? I interpreted a time and ensemble average experiment as something that can be explained by a single conformation, in this case of a coin. And the same thing is true for biomolecules. Since the experiments are averaged over many molecules and long periods of time, we have to take this into account when we interpret the experimental data. And so this is very important, and the more dynamic the system is, the more important it is. So there are many ways of dealing with this problem. Here are some papers from our own group. There are many other people who've worked on this. We've written sort of a technical review a couple of years ago by Simone Orioli and Andreas Larsen that I can recommend, or sort of a less technical review by Emil Thomasen that came out this year, if you want sort of to get some insights on this. And the work that I'll talk about, the technical aspect, was done by Sandro Gottardo, using ideas developed by many people, in particular by Gerhard Hummer and colleagues. And the basic idea is that instead of doing what most normal structure determination is, which is effectively just to find structures that explain the data as individual structures, what we will do is we'll run molecular simulations, get distribution of conformations. We will then calculate observables. Here's one example. You might calculate the measured quantity here. You would get a distribution like this red line here. You would calculate the average. This would be this dashed red line here. Someone will measure an experimental data. This can be a distance from an NOE. This can be a sex intensity. You can compare this red line to this black line, which would be experiment. You can say, well, it's not great. It's not perfect. It's not completely crazy either. Let me try to change the simulation to fit the data. So now we are using the data to the simulation to explain the data. So what we'll do is we'll do a minimal perturbation to the simulation, which would mean we will decrease the weight of this state over here, increase the weight of this state over here, and this will sort of move the calculated average from over here to over here. So instead of predicting the data, we're using, we're fitting the data, we're using the simulation as a model to fit the data. And then there are many things you need to do to avoid overfitting and all these kind of things. And these are, as I mentioned, described in some of these papers down here. And so I won't really talk much about sort of theory. I'll just give you sort of a flavor of this idea that, again, tracks back to early work from Gerhard Hummer. Although you have very different backgrounds, probably many of you will understand at least this kind of analogy, which I think is pretty good. So what we'll do is that we'll minimize this function gamma that I've called gamma by analogy with a Gibbs free energy, and it consists of two terms, a chi-square term that you can think of as an enthalpy, and this entropy term, which is an entropy, and then this fudge parameter here, theta, that behaves like a temperature. And so we want to minimize this function, and we can do that by minimizing the chi-square over here, right? But also we don't want to decrease the entropy too much. So there's a balance between this chi-square term, which matches how well are we fitting the data, and this entropy term, which quantifies how far have you gone away from your molecular simulation. So the goal is to change the weight of all the structures in the molecular simulation, but do it as little as possible so as not to decrease the entropy, but at the same time by decreasing the chi-square, which means improve agreement with experiment. And the balance between these two things is set by this temperature-like parameter theta. So here's a toy example that illustrates this a little bit better. Over here on the left, we sort of pretend first that we know nature's force field. This is this gray distribution over here. This, we sample and it gives us, this gray distribution, and we calculate the average of X and Y. This is this black point. And now we say we make a measurement of this system, and this would be the measurement. This would be this purple point. This would sort of be, the true ensemble Unfortunately, we don't have the true ensemble. We have a force field that's imperfect. That would be the red force field over here. And if we sample that, you can see we get a different distribution. If you calculate the average of X and Y, this would be this red star over here. So this would be the starting point for the work. We have an experimental measurement, which is the purple point. This is just average of X and Y. And we have a distribution of confirmations that give us an average that is imperfect. And now we'll try to fit the data, which is this purple point over here. So what we will do is that we'll take the weights of all of these different structures. We'll try to change them a little bit so we move the red point down towards the purple point. So we do that at very high temperature. We are not really allowed to change the initial force field. So we end up with a high chi-square. This is illustrated over here. And we get up at a chi-square that's above 6, which means that we're sort of 6 units away from this experiment. Or we can try to change this temperature to fit the data, but we do that at the cost of changing the weights of the different structures in the molecular simulations a lot. And this we can quantify by this parameter that we call phi effective, which effectively means how much of the simulation do you need to get rid of to fit the experimental data. And so often the curves will look something like this, where you, by essentially getting rid of a small fraction of the simulation, get a large improvement in the agreement with the data. So for example, by downweighting about 20% of the simulations and upweighting the rest, you can go from a chi-square that's more than 6 to about 2. So you pick some point along this curve. Again, this is described in these reviews, and you find some solution somewhere down here where there's a compromise between improving agreement with experiment, but not going too far away from your molecular simulation. And then you end up with a solution like this. Again, you can see now the calculated average is per construction closer to the experiment. And now you can see the distribution you get out, although we just fitted one measurement or 2 measurements, the average of X and Y, you can see the distribution of confirmations we get out is much closer to the true force field that we didn't know, we just knew the average of it, than we started out with. So by taking an imperfect force field, the red force field, and fitting it to a single set of measurements, we can get an improved distribution of confirmations. And then, there's a lot of technical details that I'm not talking about, that enables us to do this in a relatively robust way. So here's a more biomolecule sample where we did work on small angle X-ray scattering data on the protein alpha-synuclein. So Lina measured data on alpha-synuclein. We took simulations from either Mustafa's work on from Paul Robustelli, and we calculated, for example, the radius of gyration. We calculated the sex data for all of these ensembles, and then Francesco and also Mustafa did various analyses using this method that I just talked about that we call BME. And so if you just calculate the sex data from in one of these particular simulations, it does a pretty good job of fitting the experimental data, but it's not perfect. And then by changing the weight of the different frames in the molecular simulation, you can fit the data. And so what you can see is that you can get start with quite different ensembles depending on which force field or method you use to sample. You can get a much more homogeneous set of solutions, something that actually fits the experimental data. So we can use good but imperfect molecular simulations plus experimental data can give us reweighted ensembles that we can then interpret in terms of biomolecular dynamics or function. So this method BME and the code that we've developed is pretty robust. It's very versatile. Here, just some examples from the group from the last years. This I'm not going to talk about, but just to show that we can work with NMR data, small angle X-ray scattering data, in particular neutron scattering data, different types of NMR data, membrane proteins, soluble proteins, many different things, because it's very versatile. You can use your normal simulation tools and just plug it in afterwards. So as one example, I'll give you some work that Tone Bengtsen did some years ago, and this is a collaboration with Lisa Arlett's group on what these lipid nanodiscs look like. And so lipid nanodiscs, you might be familiar with, are often used as carriers to study membrane proteins in solution. And what the question we were trying to address is, what do these proteins actually look like in solution? So on the outside of these nanodiscs, there's some protein. This is the red and blue, and in the middle there's this gray thing here, which are lipids, in this case phospholipids that are sitting in the middle. I'm just highlighting one of these as in yellow just to see that they can diffuse around. So we have a relatively liquid bilayer that's trapped within these things. And then we want to address, how do they look like? This is a molecular simulation, but how do they look like in reality? And the starting point was actually something quite intriguing, which is that there happen to be both NMR experiments and small angle X-ray and neutron scattering experiments on the same system, or rather, Lisa's group measured X-ray and neutron scattering on the same system that had been studied by NMR. And so if you look at this nanodisk by NMR, I'm not showing the lipids, but there are lipids here. This is work from Ola and Week's lab. Then it sort of looks pretty round-ish. So it looks like it's more or less sort of a really spherical disk and with a lot of data. But if you do the same sort of analysis on the scattering data, it clearly a round shape does not fit the scattering data. And if you try to fit it with a single shape, it really has to be sort of ellipsoidal. So the question is, are these nanodisks round or are they ellipsoidal? And I'm not taking too much away from the surprise is that it's really a mixture of everything, right? It's a dynamical system. And it turns out that if you look at this sort of in this average way, and you look at it on average from NMR, it is sort of mostly round. And if you look at it sort of on average from scattering, it's mostly ellipsoidal for reasons that, we can discuss later maybe. So what Tone did is that she ran MD simulations of the nanodisk. She calculated NMR data. She calculated the scattering data. It fits pretty well, but there's not perfect agreement, for example, with the NOEs. Most of the NOEs are sort of below this upper bound, but you can see that there are some violations. And if you then run MD simulation, this is this blue line over here, and compare to the experimental scattering data, I'm just showing you a small part of the Sachs curve over here, you can see that the MD sort of captures nicely this dip over here. But then there's some discrepancies over here that is actually captured. If you take this, what's labeled PDB here is the NMR structure that I mentioned before. So the MD simulations really capture quite a lot of the data, but it's clearly not a perfect description of the NMR and the Sachs data. And so what Tone then did is she used this BME fitting tool that I just mentioned, essentially changing the weights of all the frames in the MD in this robust way where we avoid overfitting. And then she showed that we can fit both the scattering data and the NMR data to get out a distribution of conformations. I'm showing this here just as clustering, so you can see that there's some ellipsoidal shapes in one direction, there's some ellipsoidal shapes in the other direction, and things that are sort of more round. And so we get sort of a distribution of how not round they are. So this axis here measures on how round each structure in the simulation is, with 0 meaning perfectly round and very high value meaning very ellipsoidal. And so you can see you get a distribution of shapes going from something about as round as the NMR structure to something as ellipsoidal as this average Sachs structure. And most of the structures sort of lie in between these two shapes, these two extremes. So we have essentially an object that has ellipsoidal fluctuations between something that is pretty round and something that is very elongated, and it can fluctuate in both ways. And if you average over that and you look at it by NMR, it looks sort of round. And if you average it and look at it by X-ray and neutron scattering, it looks sort of very ellipsoidal. And in reality, it's a mixture of all of this with some specific population weights that we can estimate using this procedure. So again, we can try sort of to make sense of biomolecular data using simulations as an interpretation tool of for looking at the structural and fluctuations of this molecule. I'll just check my time here. All right, good. So from what I just talked about, we first started out by doing molecular simulations and compared to experiments. Then we moved on to this BME method where you run molecular simulations, you compare with experiments, and then you fix the simulations, but you do it sort of in a system by system basis. So we fix the nanodisk ensemble, but in ways that won't really inform us if we study another nanodisk or if we study another membrane protein. And so what we've been starting to work on is how can we use this kind of discrepancies between experiments and simulations to really improve the molecular force fields. So I'll skip through this first part relatively quickly. It's mostly published, and I want to get to the last part, and I'm running out a little bit of time. We've been using this coarse-grained Martini model, and we sort of started out by looking at a couple of different systems. First is tier 1 system. This is work by Andreas together with Lisa Lett, and then HNI-PA1. This is really mostly work from Tanja Mitak's lab, and all the experiments, not all, but many of the experiments were done by Erik Martin. And then from my side, this was Emil Thomasen. And I won't really talk about the details. What we just did in those cases, we did simulations with the Martini model. And in both cases, we found that if we compared to small angle X-ray scattering data, we found that the proteins were simply too compact. So we were looking at these flexible molecules. And in both cases, we found that the simulations were about 25% too compact compared to nice high quality small angular X-ray data on these two systems. We said, okay, it suggests that there's a problem with this force field for these kind of systems. It hasn't really been developed and tested extensively for these kind of flexible systems. So we said, okay, maybe we can use this discrepancy to improve the force field to look at these kind of systems, but in ways that are sort of transferable. And so again, this is work mostly by me, but also with work by Francesco and Mette and Giulio. And so we started looking at a whole bunch of different systems. We collected data mostly from the literature on intrinsically disordered proteins, in particular intrinsically disordered proteins that had small angular X-ray scattering data. We can, for example, run simulations of all of these. We can calculate the radius of gyration. We can compare to an experiment. You see, we get a nice correlation, but with a slope that's smaller than 1, meaning that effectively the protein is always too compact. And then, inspired by work that had been done in the all-atom work, what we did is we said, well, the proteins are too compact. Let's make them more expanded by increasing the interaction between protein and water. So we're tweaking a single parameter in the force field. Again, this is described in this published work. And by tweaking this single parameter, we can effectively fit all the data, more or less within experimental error, not quite. So we can either compare the radius of gyration, or we can compare directly with the small angular X-ray scattering data. This is a log scale. So the difference is really quite dramatic. And so by increasing the interaction strength between the protein and the water, the protein obviously swells, and we can more or less describe all of these systems, pretty well, both intrinsically disordered proteins or multi-domain proteins. Again, we have more data to suggest that this really works quite well. So we have now used deviations between experiments and simulation, and we haven't fitted the individual one by one. Instead, we've come up with a single sort of hack or fix to the force field that allows us to do, look at new systems for which we don't have data. So this works pretty well, and we're very happy with it, and we're using this method for a lot of work. But of course, it is somewhat arbitrary that we selected this single parameter, the interaction strength between protein and water, to fix the force. So we could have picked other parameters, the interaction between protein, and maybe it was something about the charges. There are many things that we could have changed. So what we are sort of instead trying to now is then can we sort of try to change all the parameters in one go. And in particular, this is work now mostly done by Giulio to say, but the first part of this work, Thea and Ramon also had key contributions to this. This first part of the work is published last year. You can look up the details. And the basic is we want to sort of look at a coarse-grained model of disordered protein where each residue is a single bead. We're starting with some work by G.T. Mittal and Rob Best, a model called the HPS model. And we want to improve this model by training the parameters of the force field against experimental observables. I'll show you how it works in a second. And then what we want to do is we want to look on self-association, condensate formation of these disordered proteins. So the goal is to have a model that's very fast so that we can simulate not just individual disordered proteins, but also interactions either between, a couple of chains or, hundreds of chains. So before we sort of started this, we just want to sort of to benchmark a couple of different models. We took this original HPS model from G.T. Mittal. The model has essentially one parameter per residue, a lambda parameter that says how sticky an amino acid is. And this came out, of other people's work. I won't talk about those details either. So there are some amino acids that are sort of, you can see these kind of hydrophobic amino acids. They get a high sticky parameters, and then some of the charged and polar amino acids to get a low stickiness parameter. So these residues will like to stick together, and these ones won't like to stick together. So this is one model. This is sort of a hydrophobicity scale based model. This is a little bit, one of many hydrophobicity scales. So what Giulio did, he just went to the literature, found 70 different hydrophobicity scales, averaged them, and this is, another set of hydrophobicity. So actually, this is the average over many hydrophobicity scales, and you can see the parameters are somewhat different. But again, with some spread in, some things that are being less hydrophobic and other things that are being more hydrophobic. So if you take this model, run simulations where essentially the compaction of the protein is governed as by these stickiness scales, we get sort of interesting, but also sort of surprising results. So if we take this model over here and we simulate variants of the protein called A1, or the low complexity domain of A1, where Tanya Mittag's group has measured the radius of gyration for all of these different variants. You can see that actually fits the data pretty well. So there's essentially a perfect match with the RG for all of these proteins. But then if you take the same model and you run these more expanded IDPs, so this is a relatively compact IDP, these are more expanded, you can really see that it underpredicts the radius of gyration, makes them quite a bit too compact. So this model works well on these proteins, but not well on these proteins. This scale over here has the opposite behavior. It works pretty well on these more hydrophilic proteins, but if you try to simulate these A1 variants, actually it predicts them to be too expanded in all cases. So we have two models. This model works pretty well on this set of proteins. This model works pretty well on this set of proteins. And this, of course, is not very useful if you don't know what the compaction is before you start. So what we did is we went back to some work we did quite many years ago, actually, that we sort of revived and updated, where we have an algorithm. And again, I won't talk about how it works, but I'll show you in a movie fashion in a second, where we say we want to optimize these parameters, lambda here, which are essentially the force field parameters. There are 20 parameters, one for each amino acid. We want to optimize them by fitting the experimental data, but not just on a single system, but across 50 different proteins. And we'll do that by minimizing, the chi-square, or maximizing the agreement with RG data from SACS, parakinetic relaxation enhancement data from NMR, and then some sort of regularize over here that I won't talk about. So we'll run simulations, we'll predict the RG and the PRE data, we will get maybe good agreement or bad agreement, and then we'll change the force field a little bit until we come up with a force field that fits all this data very well. And then we'll do all sorts of tests to make sure that we don't overfit. So we first went to the literature, collected a lot of data, a lot of SACS data with the rate of gyration of all these different proteins, the A1 variants that I just talked about, various fragments of TAR1, and then each of these are different proteins that people have been studying, disordered proteins by SACS, or these proteins over here, where there's NMR parakinetic relaxation enhancement data, or few proteins where we had both data. So we really use quite a lot of data that people have measured and published and put in databases, or were happy to send us. So now in a second, I'll show you a movie of what's going on. But before that, I'll just show you what to look for. So over here is the model that I just showed you before. Arginine is the least sticky parameter. Phenylalanine is the most sticky amino acid. So these things over here will start to move as we optimize in the force field. Over here on the right, I'm showing you example of what the results of a specific force field will look like for alpha-synuclein. So this is a specific NMR experiment, a PRE experiment, where the black is the experiment and the red is the thing that we calculate from the simulation. And for those of you who know about NMR PREs, this will show that the protein is way too compact. There's too many interactions. There's clearly not agreement. This is also seen if you look at the radius of gyration. It's clearly this average is much smaller than the experiment. And if you look at the structure, it's pretty compact. So what we then do is that we run simulation, we're changing the force fields. You can see the force field parameters are updating, and slowly you can see here, we're getting to sort of a scenario where the simulations actually predict the PRE data and the SACS data pretty well. And now we get a completely different set of force field parameters. So for example, arginine, that was the least sticky, is now one of the more sticky amino acids. You can see tyrosine over here is suddenly very sticky. Some of the hydrophobics are very sticky. And then you can see some of these polar ones, but not arginine, are the least sticky amino acids, right? So this is a very different hydrophobicity or stickiness scale than the one, this HPS scale that we used with, right? And so instead of using that parameter scale, now we can use this force field, which we fitted not just on alpha-synuclein, but on all these different proteins that I showed before. We can use that and we can start to look at how do intrinsically disordered proteins look like on a single chain basis, and how do they self-associate. So again, this is what we did. We fitted the RG data. This is just to illustrate how well we can fit the data, right? This is, the experimental data versus the simulated data with this model that we call Calvados. And so you can see we get an almost perfect agreement. These are the data that we fitted. So it's maybe not so surprising that we can, get a good agreement, but it's still amazing that with only 20 parameters, you can actually fit all these RG data. If you look at just the subset, these A1 low complexity domains, these are all the same length, but with different sequences, either different patterning or different numbers of hydrophobic and sticky amino acids. You can see we get a pretty good correlation between the experimental and the simulated RG. I think maybe most convincing, here's a set of variants that we didn't look at and at all while we were parameterizing the model. We only looked at it afterwards. And again, you can see we really predict the radius of gyration almost perfectly for these variants of the P27 protein, that again are all the same length, but where the amino acid patterning is different. So again, other models can do this, but I think it's very nice that our model that have not seen any of this data, can be trained, on all this protein and then can do this kind of prediction. So it's a pretty good model, despite the fact that it's very simple, a single amino acid, a single parameter per amino acid, plus a very simple model for the charges in the protein. So we can do many things with this model that I won't really have time to talk about. But just for example, to show that we can look in this case of a protein called FUS. This is the single chain contact map. You can see that these light stripes, these are the amino acids that like to stick together. These are sort of these more sticky amino acids, the aromatic residues, arginines. These are, kind of things that like to stick together on a single chain. If we take two chains and look at the interaction between two molecules of FUS, you can see you get a very similar pattern, right? So the inter and intramolecular interactions are very, very similar. And in fact, if you throw in hundreds of chains in the single simulation box, you can see you get more or less the same patterning of the interaction between one chain and the average over all the other chain in what is now then a simulation model of a biomolecular condensate. So following up on works by others, but using this force field that has really been trained very directly on the experimental data using the method that I just talked about. So we've used this model for many other things, but in the interest of time, I won't talk about it. Some of it is published, and I'm happy to talk about it. Instead, I'll finish here just by summarizing what I've talked about, covering quite many different topics, and I realized not in a lot of detail, but to give you a flavor on the, what I think is sort of a snapshot at least of the things that we are doing. You can either start with a specific molecule that you're interested in, run simulations in the computer in the absence of knowing anything about the experiments. And if you then want to figure out how good the simulations are, you can then do predictions of experimental observables. This can be very direct things like, chemical shifts, NOEs, small molecular scattering data. Or in some cases, you unfortunately can only do sort of more indirect comparisons to populations, for example. So this has shown us that we have force fields that are pretty good. They're not perfect, but they're often good enough to give us insight that we can then use to understand what's going on in systems. Or you can do what I talked about sort of in the middle part, this kind of inverse modeling, where you use the simulation to interpret the experimental data. Many ways of doing this. I told you about this BME method where you can run simulations and then you can change the weights of the different frames to fit the data. So using the simulation not as a prediction tool, but as an interpretation tool to figure out what your data means. This works really well if you have a relatively good, doesn't have to be perfect, but a relatively good starting point. And so the last part I talked about is how we can use disagreement between experiment and simulation. If we have enough examples that are similar, really to start to improve the parameters in the force fields in a more systematic way to try, and I told you about work on coarse grain models, but the same ideas can be applied for more detailed models, where we can really try to sort of fit the parameters in a molecular force field to do transferable model on systems where you don't have as much data. So I'll finish by acknowledging the people that did work. I mentioned most of the people along the way. The work on P27 was done by Joao, and the VAC 1 was done by Simone. A lot of the SACS work, at least at the moment, is done by Francesco. The Martini work that is mostly done by Emil. Some of the FlipAce work was done by Young, who's not in the picture, and Felix is taking that over. And the last part is in particular work by Judu to say who's continuing this work from the group. And so with that, I'd like to thank you for your patience and your interest, and I'm happy to take questions for you.
Thank you very much. Now we're open for questions. Carlos, would you like to start?
Yes, I can start. I think you almost already answered my question, but still, when I listened to the middle part, I was wondering what is the meaning for the simulation when you are taking part of the simulation out or just weighting down the certain parts of the simulation. What's the meaning of for the simulation? Is that the force field is wrong, or is just you are missing sampling enough time, or I don't know if I explain myself.
No, I think I understand, and it's a very good question, something we discuss and think about a lot, and we could talk about this for a long period of time. I'll try not to do that. So first of all, often we don't know whether, when you get discrepancies, whether it's sampling or the force field. So we try to do as much sampling as we can. So this is one of the origins of disagreement, of course, is lack of sampling. Another thing that I didn't talk about, that's something that we work on quite a lot, is also how you calculate the experimental observables. If the experimental observable is a SAX experiment, right, you need to calculate the observables in a way that are faithful. So, how you deal with the solvent layer, for example. If it's an NMR experiment, you have to do the averaging in the right way. If it's a scalar coupling, use the right Karplus parameters, assuming that Karplus models are correct, all these kind of things. So, and then if you ruled out that it's sampling, and if you ruled out that it's due to the way that you calculate the observable, then the major limitation is the force field. And clearly, the force fields are not perfect in the sense that there are many systems where even if you do long sampling and you calculate observables, you don't get perfect agreement. So then what, sort of on the more philosophical level, what the role of the force field is sort of a sort of model, and you're sort of, that has some free parameters, which is effectively the population of the states. And those parameters we don't know. So the analogy that I like to make, it's sort of not a completely, direct analogy, but it's sort of at least semi-correct, is, when you fit enzyme kinetics data, right? You measure velocities and substrate concentration, you have the Michaelis-Menten model, and there's some free parameters, right? And you fit those free parameters, right? You fit a Kcat and a Km, and that means that the model is not predictive, right? The model, you're fitting the data, but now you have a model, and that model allows you to say something about the system that is more than what you could do by just looking at a table of numbers, right? So now the model that we're using to interpret the data is a whole simulation with the weights of each frame. And that is a very complicated model, right? If you have 100,000 frames, we have 100,000 parameters. And so the key objective is to try not to fit 100,000 parameters completely freely, but to change them as little as possible to explain all the data. And so you might, people who do SAX, they might, fit a P of R distribution using the SAX data, and that the prior for the P of R might be, something, random, or it might be a flat P of R distribution. What we'll say is that our prior, before we've done the experiment, the best guess at what the system looks like is the molecular simulation. So this is the role. This is the best guess before we've done any simulation. This in the Bayesian framework is what we call the prior. And now we measure some data, and now we update the prior, we generate a posterior. This is after we've done the experiment. This is the best guess of what the system would look like. It's not guaranteed to be perfect, but it's better than the prior, right? Better than before we did the experiment, and it explains the data. So at the more philosophical level, it's really a model, like many other modeling tools, to interpret the system that you can then move on and say something about. Does that make sense?
Yeah. Yes, I'm experimental, so I'm trying to have a feeling of when to believe the simulation. It's like in the first part, when you have some experimental data, when you can compare the simulation with the data, I mean, yeah, if it's the same, you are happy, right? But if you don't have that experimental data, how I mean, I guess you still don't know if you just believe the simulation or not, but that's what I'm trying to get a feeling of.
Yeah, no, I mean, it's a good point. I don't think that there's a simple answer. If we take one extreme, which is the models that are used to determine most protein structures in the PDB, let's say from NMR, right? Something like Cyana, right? The force field that is used in Cyana is incredibly simple, right? It's a little bit more updated now, right? But it used to be, bond lengths, bond angles, and hard sphere interactions. So if you run a simulation of, ubiquitin with a Cyana force field, you'd get, nonsense, but it would not look like ubiquitin. So this is a force field that, this is a force field that nobody would trust in that sense. And now you plug in all your NOEs, you do your refinement in Cyana, you get a structure, now we trust this structure, right? It's based on the data. Right? And so we start with a very simple model that is very wrong. We plug in some data, we get a solution out that we trust more. But it might not be perfect, right? Not all in mass structures are perfect, but it's clearly better than the starting point, which was complete nonsense. So if we have a force field that is better than the cyana force field, right? If we have an all-atom MD force field, it's clearly better. So the first guess at what the structure would look like is a better guess. Even if it's not perfect, it means that by putting in, if you put in more data, you get an even more robust solution. But often in practice, what we can do is that we can use less data and get a relatively robust solution. So by having a better guess to begin with, right, we don't need so much data to get something that at the end is relatively good. But putting an absolute number on the trust is very difficult, right? And it really depends on the questions that you are trying to, to come out, right? If you want a probability of, the atomic accuracy, or whether you want to say, well, I want to understand, is the distribution of Rg, accurately described? These are very different questions, and they come with different levels of certainty depending on what you're doing.
Thank you very much. It was really, really a nice talk. Yeah. I don't know if I understand everything, but yeah, the little that I get is really nice.
Thanks. Yeah. So this topic is something, this last topic is something I'm very happy to discuss also, more also later offline if you're interested.
I have a bunch of questions. One of them is, when you're looking at the lipid selectivity of the transporter, that requires fairly accurate description of the interactions between the lipid and the protein, right?
Yes.
So what force field does that for you?
Yeah, so it's sort of, so most of the data that I showed is coarse-grained from the Martini model, which is obviously, doesn't have a lot of chemical detail. On the other hand, of course, the Martini model is known, has been parameterized and tested a lot to look at lipids. But of course, there is a lot of chemistry that we don't capture the details of, both on the lipid side and on the protein side. There is a problem. This is related to the discussion before. I said that we capture the selectivity. We don't really know exactly what the selectivity is, certainly not for binding. Right? What has been shown experimentally is if you do turnover, if you measure ATP hydrolysis of the ATPase, how does that depend on the presence of different lipids? That's very difficult to translate into a specific number of, the binding free energy. Obviously, it's much more complicated than that. So this is one of these examples where we can't really do a direct comparison. So we have to do something, more rudimentary. I think, and again, we're not 100% sure of this. We think that there's more selectivity in the actual biological system than what the simulations capture. So the difference between the different lipids in experiments appears to be bigger than in the simulations. And I think that there are two explanations of this. One is what you just alluded to, that the simulations aren't good enough to capture this. I think this is part of the answer. But I think the other part of it is that there's additional selectivity that comes from the part of the process that we're not studying. Right? So we study the first initial move of the head group up sort of to the middle of the protein. And then there is a conformational change induced, occlusion, and then, opening to the other side. And there's probably some selectivity in that process that we're not capturing because we're not studying that process. So one of the questions that we are trying to address is, where does selectivity come from? And what I think the conclusion is that selectivity comes not just of binding of the substrate, but also of the ability of the substrate to elicit the conformational change, for the second half of the process. But again, this is sort of a negative, this is negative evidence, right? We don't see all the selectivity. And so this probably means that there's more selectivity going on later. But it could also be, the fact that the model just isn't good enough to capture this strong difference. We've tried to do this with all atom MD. It turns out to be even more difficult to get converged free energies of lipid binding with all atom MD. So we've tried quite hard and have not been able to get numbers that are sufficiently converged that we can report meaningful statistical differences.
So in order to get to this convergence issue, is it useful to draw thermodynamic cycles like the ones you showed? So if it adds
Yes.
up, then you're happy?
Yes, exactly. So what we've done is that we've essentially mutated one lipid in the binding site and a lipid in the bilayer. We sort of, we take a PE and a PC lipid. We take the PE lipid and make a PC, and we take the PC and make a PE. So the chemistry preserves, so we're essentially swapping, lipids with alchemical free energy changes. So we get the free energy, relative free energy between the bilayer and the binding site. And we can do that, for each pair. And then we can ask, does the thermodynamic cycle add up at the end? So this is the control. And for the free energies that are showed, which was from coarse grain simulations, they add up very, very nicely. If we do this with all atom MD, they don't add up perfectly, which tells us that there's something that, that there's errors in our calculations. We're not converging the free energies, even with, quite substantial sampling. But they're just, they're big, relatively big alchemical differences and, complicated systems, so difficult to sample.
Right. I don't know if we have any more questions coming from YouTube. While those come or not, talking about sampling, when you say extensive sampling, what is like an idea of how long should one run in order to get something that people are willing to believe?
Yeah, as much as possible.
Within a lifetime.
Yeah. I think that there's no single answer to that question. And I think it really depends on the goal of what you're trying to do. So for example, if your goal is to be in this kind of very predictive mode, I think running extensive sampling is extremely important. If you want to do this more integrative stuff, extensive sampling is also really useful, but actually you could also do some things if you don't have perfect sampling, because then the fitting to the experimental data helps in restraining it, right? So this is something we and other people have looked at, right? If you do the reweighting the way we do it, you need to sample the relevant states. But even if you don't sample them exactly with the right populations of the force field because of imperfect sampling, you can still do quite a bit of useful stuff. The way that I like to talk about it in terms of when we start to in the absence of this, if you want to do predictions, is that when you can start to calculate error bars that are meaningful, then you're in a good shape, right? So I think our experience is that early on you can't even calculate errors, right? You're so unsure about your sampling that you don't even trust your error bars. And when you don't trust your error bars, there's no way that you can trust the numbers. And of course, the error bars also have errors, right? And so, of course, in the end, it becomes sort of a, stacks of tortoises all the way down. But at some point, I think when you've sampled the processes to some extent, at least the error bars that you calculate, the errors on the observables start to be meaningful. They might be large, but then I think that's when you can start to trust things, right? Then you can address, can you say something about the difference? Is the free energy difference between this state and that state bigger or smaller than some number? Is the RG, bigger or smaller when you make a mutation, or all these kind of things, right? But until your error bars are meaningful, then you really haven't sampled enough.
I agree.
And then, of course, it depends on the, you can have convergence of some properties, but not of other properties, right? So if you care about the compaction of a protein, you can often get relatively good estimates, even if you, in the all-atom world, even if you haven't, for example, sampled all the details of the local secondary structure. They might be coupled, so of course it's not completely strictly true, but overall some properties are easier to converge than other properties. But surprisingly often the answer to your problems is just to run more, which of course is very frustrating.
I agree. And my last question has to do with Calvados.
Mm-hmm.
When I saw the paper, I wanted to use it to sample what a peptide would do. It's a disordered peptide. It's amylin, actually. And we're looking for whether different variants of amylin show different conformational ensembles. If some would be more prone to form fibers, amyloid fibers, or not. or if they would be happy to bind ions. Do you think it is the right tool to do that, or should I go to all atom descriptions?
Yeah, or something in between maybe. So I think the model is extremely good to many things, but there's several limitations, right? One limitation is that it's one bead per residue and there's no local potential, which means that there's no secondary structure.
All right.
But there's some people that are working this, right? So if you want to address things like formation of beta sheet-like structure, right, this is difficult, right? You can look at compaction and expansion, you can look at transient long-range interactions, but questions about changes in local secondary structure, the model just cannot do as it stands now. And then in terms of ions, right, there's no water and there's no explicit ions. All the electrostatics is essentially guided by each charged amino acid has a full charge, and they're screening by a sort of implicit solvent, the bi-Hugel term. So it's very simple. It actually works surprisingly well for looking at salt effects of compaction and charge patterning, but it clearly doesn't have details about, binding sites for specific ions. Then at the other extreme, you have, all atom MD, but convergence even of a, 30 residue peptide can be very, very difficult. And then for some questions, I think this Martini model is extremely good. And, this tweak that we've done that enables you to study flexible peptides and IDPs, can at least for some questions be useful, right? There's also no secondary structure, but at least there's a backbone and there are specific ions and there's a little bit more chemistry in the side chains. So this is sort of an intermediate step. And, one approach that we haven't formalized, other people have been trying to do things like this, is that, maybe you can bridge across these resolutions in different ways, right? So you can sample structures with, with one of the models, or you can go fine-grained and you can sort of fill in the details with the other one. So this might be some of the approaches to do. But I think, if you want to look at things like secondary, structure, ion binding, I don't think there's any other tool than all atom MD out there to do these things.
All right. So there being no more questions, thank you very much for your talk and for the discussion. It was a pleasure to have you.
Thank you very much, everybody, for coming and for your questions. And if there's things that you want to follow up with or ask about, just send me an email. I'll be happy to try to answer your questions at a later stage. Thank you very much and have a nice day.
Bye-bye.
Thank you
Happy
very much.
dinner.
Bye.